All right, welcome to Unit 2, where we'll talk about food's nutrient parts. We're going to start off talking about macronutrients. These are your carbs, your lipids, your proteins, what we call energy-yielding nutrients, which means that they end up yielding calories, or they're used to produce ATP, or energy for our cells. And then we'll take a look at the micronutrients shortly after this. So we'll start off with carbohydrates. These are literally hydrated carbons. So you have a CH2O molecular structure with each one of these carbohydrates. That's the ratio in every single carbohydrate that we're going to talk about. Overall, these yield 4 calories per gram. And it's important to know that these are your primary fuel source during exercise, while lipids or fats are going to be your primary fuel source during low intensity activities such as resting, sitting, and sleeping. When you start exercising, you're going to tap into more of your carbohydrate stores. Overall, the Institute of Medicine recommends that we consume about 45 to 65% of our calories from sources of carbohydrates, and then we'll talk more about the quality of the sources as we go further. But here are a couple of structures on the slide here. We have our simple carbohydrates, which are our monosaccharides and our disaccharides. Now we have 3 different monosaccharides. When you pair these monosaccharides together, you get di, which means 2 saccharides. Then we have complex carbohydrates. These are your oligosaccharides, where you have 3 to 10 monosaccharides stuck together. And then polysaccharides, which have more than 10 of these monosaccharide units. These are typically chains of glucoses. We can take a look at these simple forms of carbohydrates here. On the left-hand side of the screen, you can see our monosaccharides, and all of these share the same molecular structure of C6H12O6. That's 6 carbons, 12 H's, or hydrogens, and 6 oxygens. It's just how these are put together, which is different. Monosaccharides are going to be absorbed in the small intestine. They're not going to usually make it to the large intestine, where a lot of our bacteria are. But this is their form in which our carbohydrates are eventually going to be absorbed, and that's a key point with the monosaccharides. But in nature, we will find our simple sugars as disaccharides, coming from sucrose, which is just glucose and fructose put together, or lactose, which is galactose and glucose put together, and then maltose, which are 2 different glucoses put together. And each one of these has enzymes in our gut responsible for breaking them down. So you have sucrase. If it ends in ASE, that must be an enzyme, right? So sucrase breaks down sucrose. Lactase breaks down lactose. And of course, maltase will break down maltose. Maltose, it's created when starch is fermented by yeast or enzymes such as bread or brewed beverages. Lactose is more of a milk sugar. Sucrose is what we commonly would see with table sugar. It's found in plants, fruits especially, sugar cane and honey. About 3/4 of people have primary lactose maldigestion after ages 3 through 5. This is caused by a reduction in the production of lactase, the enzyme to break down lactose. And if serious symptoms can occur, then we will end up calling it lactose intolerance. Now, this is estimated that 3,000 to 5,000 years ago, a genetic mutation actually allowed for certain groups, mainly those in Northern Europe, to keep producing lactase. Today, African Americans, Asians, and Hispanics are most at risk for lactose intolerance. So here we have our complex carbohydrates. At the top of the screen, we have those oligosaccharides. It's 3 to 10 sugar molecules in length. These are found in dried beans, peas, and lentils. And their oligosaccharides actually don't get absorbed in the small intestine for the most part. And they act as prebiotics, should be pre, not pro, since we can't break them down. Prebiotics are feeding our friendly gut bacteria in our large intestine. Polysaccharides include starch, glycogen, fiber, and cellulose. And the interesting thing about starch is we have chains of glucoses. You can see these chains of glucoses on, towards the right side of the screen here. And notice that the structures can either be branched or in a long straight line. The branched ones are called amylopectin, and then the linear ones are called amylose. Now, the difference between the two is that amylopectin, the branched one, is broken down much more quickly than the amylose, and that's because enzymes can remove the last glucose at many different sites on the molecule at the same time, and then it can be somewhat eaten up more quickly than can an amylose or a long chain molecule. Now, amylopectin is kind of similar to our body's stored form of carbohydrate, which is glycogen. And we store glycogen in our muscles, and that tends to be a primary fuel source during exercise, especially higher intensity exercise, and then also in the liver as well. And when we have low blood sugar levels, the pancreas will secrete the hormone glucagon, and it tells the liver, hey, why don't you break off some of that glycogen, that liver glycogen, and you can spit it out and we can have some more blood glucose. So it's a way that we can normalize or regulate our blood sugar levels. Now, wheat flour contains higher amounts of amylose. Cornstarch has higher levels of amylopectin. Now, would it make sense that a diabetic or pre-diabetic would be told to have more amylose than amylopectin, maybe more of the wheat flour and less of the cornstarch? And that should make sense, right? Because a pre-diabetic and a diabetic wants to limit the rate of absorption of these carbohydrate sources. By doing so, they're limiting the insulin response, other hormone released by the pancreas, which has the role of reducing blood sugar levels by acting as a key to the cells and allowing glucose to be stored as fat or be stored as glycogen or be oxidized inside the cell. So then we move to fiber. Now this is defined as an indigestible form of carbohydrate. These are not bound by alpha bonds like our starch. These instead are bound by beta bonds. You think we're the alpha species, right? So we can only break down alpha bonds. We cannot break down beta bonds, which means that these don't get digested. These remain intact all the way through the small intestine, and they make their way into the large intestine. And they can actually feed our beneficial bacteria, and that gives them a major role in our diets. The book describes functional fiber as this isolated indigestible carbohydrate that has beneficial effects on humans. And this is really split into two different forms, soluble fiber and insoluble fiber. The soluble ones are dissolving and swelling when put into water. These are fermented by bacteria in the large intestine, so this is food for bacteria. And these become gel-like, and they actually slow the rate of absorption in the small intestine by attracting water into the GI tract, and they can end up reducing our blood cholesterol and control our blood sugar levels. So those with blood sugar issues are often told to include lots of fiber in their diet, especially when they have more simple carbohydrates in a meal. They'll say if you're going to have the cake, have it with some beans and nuts on the side. The insoluble fiber, a little bit different. This one does not dissolve in water, but instead it will add fecal bulk, and it acts as a natural laxative. It speeds up what we call transit time of food through the GI tract. And you can describe transit time as the time it takes for your food to get from plate to bowl. Now, the function of fiber, it's got many of them. It actually has its hand in many different chronic illnesses. So we already said that it can maintain healthy bacterial flora. Believe it or not, when bacteria consumes fiber, oftentimes it will excrete short chain fatty acids like butyrate. And that butyrate can be consumed by the cells lining your large intestine. It could actually be food for the cells of your colon. In addition, fiber can reduce your cholesterol levels, and that's because one of the functions of cholesterol is to form bile. Now, bile is released, it's actually created in the liver, stored in the gallbladder, and it's released in response to consumption of fat. And once it enters into the small intestine, it will emulsify the fat, similar to shaking up your oil and vinegar salad dressing. Once you shake it up, all of the olive oil, it will sort of split apart into many different bubbles of it. But then if you sit it back on the table, you guys know what happens. It ends up coagulating back together. What bile does is it keeps those little bubbles of fat apart from one another. And by doing so, it will allow your enzymes to attack in many different more binding sites for that fat. So it speeds up the rate of fat breakdown or digestion inside that small intestine. So what does this have to do with cholesterol? Well, fiber actually binds to bile and brings it with it all the way into the large intestine. So by consuming fiber, we are actually reducing our cholesterol levels in the body. And this is because bile typically gets reabsorbed late in the small intestine. It gets reabsorbed, gets sent back to the liver to be used in various ways. So when we consume fiber, we are reducing the net amount of cholesterol that we have in our bodies. And this ends up showing up on blood lipid panels as having lower, say, LDL levels. Now, fiber can also increase the glycemic, decrease the glycemic index and load, and this does so by slowing the absorption, as we talked about before. It acts as almost a lattice network over our enterocytes down the small intestine and reduces the rate of absorption of glucose, and therefore you're not going to have that same insulin spike. Fiber has been shown to decrease your cancer risk as well, especially cancers related to the colon. Colorectal cancer is the number 3 cancer in America in both men and women, and also prostate cancer as well. This extends even to breast cancer. It's been shown that women who have less fecal bulk have a greater risk of breast cancer. And by reducing transit time, you're moving the food through your system, and essentially less bad things can happen if it's there for a shorter period of time. The last one here is to reduce systemic inflammation, and this one is big because it's a characteristic in every single major chronic disease. Obesity can be defined as a chronic inflammatory state. So the point is we need more fiber, but how much do we need? Well, for men, they recommend 38 grams. Women, 25 grams, but you can go by the 14 gram per 1,000 kcals that you're consuming. The daily value used on food labels is 25 grams. So shooting for, say, 28, 30, or even more is going to be beneficial. I encourage you to watch the Dr. Greger Nutrition Facts video on how much fiber that we actually consume, and I'll post that after this video. And now we make our way to sugar, and the average American consumes way too much sugar, 20 teaspoons of added sugar per day. Now I say added sugars because this is really what we care about the most. Now added sugars are defined as caloric sweeteners added to foods during processing or preparation before consumptions. This is not the sugar naturally occurring or found in your fruits and vegetables. This is the sugar in your fruit juice, but this is not the sugar inside the fruit itself. So added sugars are what we care about the most, or free sugars as you might hear it called. The NHANES survey shows, which is a big national nutrition examination survey, it shows that 14.6% of our total calories that we consume is coming from added sugar, and that is way too much. 60% of that comes from sugary sweetened beverages. That's our main culprit, but also grain-based desserts and fruit drinks as well. Now, the World Health Organization, as you'll see in the next slide, recommends reducing our intake below 10% of our daily calories, and ideally, for added health benefits, below 5% of our daily calories. Now, here's a question for you. How many grams of added sugar equals 5% of a 2,000 calorie diet? Well, we have calories and we have grams, and we said before that there are 4 kilocalories per 1 gram of carbohydrate. Divide 2,000 calories by 4 to see how many grams that we'd be consuming across the day, and that would be 500. So 5% of 500 is 25. So 25 grams of added sugar equals 5% of a 2,000 calorie diet. Here is that World Health Organization guidelines for sugar intake in 2015. You can read through this. It defines what I've just talked about. Next up are alternative sweeteners. These are synthetic chemicals that make things taste sweet without the burden of these extra calories. But remember that the hormone insulin is among the most important variables to our health. As I said before, it's released by the pancreas to allow blood sugar to leave the blood and enter our cells, where it could be used as energy or stored as fat. Insulin is our body's major fat storage hormone, and in too high amounts, it can lead to all kinds of diseases, including heart disease, certain cancers, and of course, what it's known for, diabetes. But what causes insulin to be released? Now, certainly high blood sugar levels will do the trick, but there's evidence to suggest that it's also released in response to sweet taste. Alternative sweeteners might do the trick short term by limiting caloric intake, but long term they may create expectations in the brain to crave more sweet foods, which can actually increase our sugar intake outside of this alternatively sweetened meal. Also, there's growing research on the effect of these sweeteners on our gut bacteria. It might actually negatively affect the composition of our bacterial flora, and that in itself can have significant health implications. Another one, these are often used by people who want to lose weight, and the evidence is extremely mixed on whether consumption of alternative sweeteners is an effective method to do so. The bottom line on alternative sweeteners is to proceed with caution and to try to reduce the amount necessary. If you go without sweets for a few days, your body will adjust to not require as much. This means that we can wane our dependence on sweets, and that's a very good thing. Sugar alcohols contain less calories than sugar itself, 2.6 calories compared to 4. These are often used by those trying to lose weight as a substitute for sugar, and despite alcohol in the name, these don't contain the ethanol that gets you drunk. These are found naturally in fruits and vegetables, but what we buy are mostly produced industrially. They often end in ITOL, like sorbitol, malitol, erythritol, xylitol, and these are very low on the glycemic index, so they don't increase insulin levels. They also won't cause tooth decay like sugar will, and they may actually feed the good bacteria in the gut, similar to dietary fiber. So a much better choice for a non-sugar substitute is a sugar alcohol. And over here we can take a look at the sweetness factor within the sugars, the sugar alcohols, the alternative sweeteners. You can see alternative sweeteners have a much greater amount of sweetness per unit than sugars or sugar alcohols. So what if we want to quantify our blood sugar response from these carbohydrate sources? Well, You've probably heard of the glycemic index, and this serves as an indicator for how quickly an individual food can be digested and absorbed and show up into the bloodstream. And the way that researchers do this is they give 50 grams of a carbohydrate in thousands of different foods, and they test for the blood sugar response. So they take 50 grams of, say, watermelon worth of carbohydrate, okay, no matter how much watermelon that is, and you measure the blood sugar response in somebody that consumes it over the next 2 hours or so. You look at the area under that curve of the area under the spike that you'll see, and you can put that out of 100. You can scale it to a scale that's recognizable for everybody to see. But the problem is that 50 grams of carbohydrate in a carrot or watermelon means that you'd have to eat a lot of carrots or watermelon. So the glycemic load was created, and this is thought to be more useful because it's more of a practical assessment of a food's impact on the blood sugar. And this is created by multiplying the glycemic index by the number of available carbohydrates in a regular human portion. So it's more practical in that way. And then it's divided by 100. And you can see what constitutes high, medium, or low in the chart on the screen. In addition, there's the food insulin index. Remember, insulin is really what we care about the most here. We're trying to get an implication from the blood sugar response to tell us, well, what's the insulin response? So for the last 20 years, largely with a group in Australia, the food insulin index has been looked at, and there are many different responses by many different whole foods or even meals. In this case, they can combine protein, fat, and carbohydrate sources and look at the net effect on insulin. So it might even be more practical than the glycemic load. And the idea here is to be an added tool for those with type 2 diabetes to reduce what we call postprandial, or after a meal, hyperinsulinemia, or too much insulin in the blood, and improve insulin resistance and beta cell function. Insulin resistance is when our cells are not responding as they should be to insulin, and the beta cells of our pancreas have to produce more and more and more and more. So this places an extra burden on our pancreas to do more work over time. Eventually, those beta cells can call to life, and that is a late-stage type 2 diabetic. Over here we have a list of the glycemic index of common foods, and you can see maybe where your favorite foods line up. And it is possible to have a low glycemic index but high glycemic load food, and vice versa. And you can see that on this chart as well.